Obrigada, Júlia.
Thank you very much, Julia. You don't know how it is hard to talk. You're doing very well. I don't know if you're aware, but I'm used to microphone and to talking in public. We know you're nervous. And my son, he studies medicine, and he in his presentations he's very nervous, and he has these sweating episodes. And so congratulations to you all. And following up with the program, we invite, as a chair of this Spinal Foundation for Translational Research and Partnerships, to accelerate the technological development of biological products. Tenure professor of the Federal University of Rio de Janeiro, Leda Castilho. So my son does not study there at your Federal University. She's a doctor in biochemical engineering by the German Research Center for Biotechnology and Technical University in Germany. And she's master, has a degree in chemical engineering by the Federal University of Rio de Janeiro. She's experienced in biopharmaceutical engineering, acting in cloning and protein expression in mammal cells, with the cultivation of animal cells and purification of biomolecules, with an emphasis in the development of advanced technologies for the production of biopharmaceuticals, inputs for tests, diagnostics, and vaccines. And in the Federal University, she works with the chemical engineering at Copy School, where she works with cellular studies. She is also a professor at biochemistry school. And as lecturers in this panel, we call the leader of the division of CNPEM and Sandra Dias. She's going to make the presentation. National Center for Research in Energy and Materials, CNPEM, cutting edge science to tackle global health challenges. She has a PhD in biomolecular physicist in the University of São Carlos, in Federal University of São Paulo, and she has a master's in biomolecular science in Federal University of Campinas, and she is graduated by the same institution in biology. She concluded her PhD in the veterinary school of the Cornell University, and she works with biomolecular technologies and biochemicals and structural biology to clarify the complex interactions that happen in the progression of tumors. The Vice Director of Innovation of Bio-Manguinhos, Ricardo de Godoi. And with the presentation, Prototype Plant. He's a doctor in science relation in the pathogens and host studies with the Federal University of São Paulo, and also works with the scientific computer, and also works in biology at the Federal University of São Paulo. He works in biogenetics, epidemiological, and digital health and data science, with an emphasis on human genetics, and working especially with population genetics, epidemiologic genetics, and studies of association, binding, and analysis of complex segregation. He worked as a vice leader of the technology of information at Fiocruz, and in the coordination groups in the pandemic of the COVID pandemic at Fiocruz. He was coordinator in Rondônia, the Fiocruz branch, and coordinator in Fiocruz headquarters, and also works in the epidemiological laboratories in the Amazon. And for the online presentation, we invite the Director of Development of Bharat Biotec, Raches Ella, who is going to carry out a presentation for infrastructure to accelerate innovation in vaccines. He's a doctor in the University of Johns Hopkins University, and he has a MBA in Harvard Business School and Harvard Biotec, and he leads the development of products focused on the breakthrough and development of vaccines and therapeutics. So I pass the floor to Leda Castilho. So good morning, you all. Thank you very much. It's very good having this full room for this panel. So as the majority of the audience are Brazilians and the Zoom participants have simultaneous interpretation, I will speak in Portuguese and also to stimulate that you may ask questions and that we have a fruitful conversation here. And I'm going to pass the floor to the panelists, and then we will have a Q&A session at the end, and the production team will send me the questions made through Zoom. So, but I also understand that people can also ask people who are in the room in the audience. So you can send through the application and also in-person questions. So our table today has as a theme foundations for translational research and partnerships to accelerate the technological development of biological products. And this is a very important theme for Brazil and for the world. The immunobiological is very important for the human health, and it's a moment that is relevant that we are discussing this because the ecosystem that and myself who works as the Federal University and the cellular cultivation laboratory for the production in biologicals using animal cells. In these years I have been working with this field in Brazil, I see that we're finally having the innovation sector in this area and the biologicals and gaining momentum and a concrete drive, which leads to the development in Brazil through the whole country, because we have been developing, having developments, but mostly regarding foreign technology. And we see this growth, and these are the consequence of actions that were taken in the past. So in 2007, 2008, under the leadership of the Development Bank and the Ministry of Health, the implementation of public policies and the Development Bank especially identified at that time something that is still real, and we can see some improvements, but it's a deficit in the biologicals and the health field, and they identified that was important to be developed in Brazil, and still currently nowadays it represents half of the expenditures with medication in the country, and we see that we have high value there. And it's important that the public policy resulted during this time in the implementation and deployment of the infrastructure for biologicals. So in this area, we are not only an importer or a packager or a manufacturer for the box and the packaging to distribute. So we have plants here producing biologicals in a greater rate, and then in the synthetics field, which I think that the great majorities are imported, especially from China and India. So in the biological area, these public policies from those years had unfoldings in a way that was very essential, and the partnerships of PDPs for scientific development allowed this development happen in the private sector, but also in the public sector. And Bio-Manguinhos is a great example of this. And we see several partnerships being developed for the unified health system. And this resulted in the creation of infrastructure, expertise, and plants for the manufacturing of biologicals in Brazil. If we can recall, we see that we have this kind of plant in Brazil, allowing that in a emergency sanitary crisis. We saw that what happened in the COVID pandemic. We see that we could have the finished product due to this plant that were already being deployed in Brazil. So otherwise, we would be dependent on imports. So this production in Brazil also results of transfer technology from foreign companies. And myself, as a researcher in this field, I would like very much to see the development of technologies that are national and produced in the country, uh, for us to be able to look at the Brazilian problems, and we could be more cost efficient. And I see this with very much optimism that this ecosystem is gaining momentum and is increasingly important. And I would like to mention 5 examples, and we're going to see 2 of them here. And the third that I would like to mention from these 5 examples, I'm going to very briefly comment on them, but on the Federal University, we started in the beginning of the year to build a pilot plant to produce biologics and recombinant proteins, and to quality assurance, stability studies for pilot batches for phase 1. This is supported by CNPq and with the funding by SPAPERJ, and we have the certification. Quality assurance is in a advanced deployment phase, and we imagine that this trivalent vaccine that is being produced is going to be produced in Rio de Janeiro, in the Fundão Island. And this first batch, we took our team and equipment for facilities in São Paulo, so we could produce there because our clean rooms were not operational yet. So this is a first example, and we will have a pilot plant in the Federal University of Rio, and also in ESP with Botucatu, and with FAPESP as well. We are installing Cevasp, which is also a pilot plant with CMO and CPO, and they have a company in São Paulo called Bio-Optimize, which are very young people that founded this company, and they established facilities of CRO, and they intend to build a plant to be a CMO. And we have these 2 examples that we're going to see here, which are the LMBIO in Campinas, in the CNPEM campus, and the Bio-Manguinhos with all this structure. We also have this pilot plant, and Ricardo is going to explain to us. And we also have Dr. Ella from Bharat Biotec to talk about his experience in India. So I thank the audience, the speakers, and I pass the floor to Dr. Sandra Dias from LMBIO. Hello, good morning. How are you? Can you hear me? Okay, it's a pleasure being here today. I thank you for the invitation. I would like to start saying that I'm here today very proudly representing our director, Maria Ruda, who we call as Guda. And I'm here on her behalf and the other directors and general director to talk about our institution, the CNPEM, the National Center for Research, and also to talk about the LMBIO and how we believe that we can contribute to this network of research and development in this area.
Realmente é desafio falar olhando para essa tela de ladinho aqui. Eu vou tentar.
It's a big challenge to talk about this subject with such a small screen. I can hardly see what's on that screen. Anyway, I'll be right here. I'll be standing right here if you don't mind. It's a lot better, and then we don't get stuck here. So I don't know if many of you know, but this is our campus, the National Center for Research in Energy and Materials, the Brazilian Center for Research in Energy and Materials. It's a very large campus, 530,000 square meters of surface area geared to research and technology. We have today 4 national laboratories, about 900 employees, 500 that are not employees, of trainees, postdocs, interns, and other kinds of associated members. What is the mission of CNPEM? It operates through 4 axes, main axes. We have open laboratories for external users. They can, on the internet, we have a page in which anybody who's interested in using those facilities, open facilities, can apply and send projects, and once they're selected, they can come and use the facilities. For academic users, it's free of charge. We have our own research and development that it's done in 5 thematic areas. We develop research in agriculture, health, human health, energy and materials, and environmental issues. We provide support to innovation, our own research, and also research that's done through partnerships with companies. And we have a strong mission in education, training, and outsourcing. This map, we like to use it. It's very provocative. It talks about Sirius, our source of syncontrol. I don't know if you know that, but we have about 80 sources of low synchrotron in the world, going from the 4th to the 5th generation. Has to do with the brightness of the source. The Sirius is, together with other 2 laboratories, it's the only, in the case in Latin America, it's the only source of blue synchrotron. But Cirrus and other laboratories are the only 3 laboratories, 4th generation laboratories in the world. We have Max 4 in addition to Cirrus, Max 4 in Sweden, and we have this and Godoi, which is a multi-user European laboratory, in Cirrus here in Brazil. So there are only 3 4th generation laboratories that are the most modern laboratories. Cirrus is one of them. It's a milestone that everybody should be aware of, so we can take pride in it. We have the only laboratory in Latin America. It's one of the 3 most powerful labs in the world. We're finishing up phase 1 implementation of light lines in Cirrus. We're going to have 14 light lines operating, 7 are operational already, and another 7 are being commissioned or installed and being built. Now, on the nanotechnology laboratory, develops research in nanobiotechnology, nanobiomaterials, and devices. I brought you this information because they work with nano and micro manufacturing. A lot of the diagnostic kits that were presented here, I understand that it's obviously in your interest. They can produce by them. They have partnership agreements for that kind of development. We have the biorenewables laboratory. If you want to look into biodiversity and to find new enzymes and come up with microbiological plants and deliverables, or to convert biomass into a series of molecules like biofuels and other chemical molecules based on chemical molecules with the idea of producing biorenewables. And we also have a science school. It's in its third year. The science school has a model that's very unique. All the students selected have full support, full financial support to be able to study at that school. They have housing, lodging, and it works with a teaching model that's not very common in our universities because it's based on the development of projects. And finally, that will be our fifth laboratory, Orion. It's biocontainment, maximum biocontainment laboratory. We're going to be able to work with level 3 and 4 agencies and that institutions. That laboratory began to be built, and it will be added to CIRUS. And of course, in this laboratory, it will be a large laboratory, large area. It will be possible to develop all kinds of studies related to infectious diseases. And these studies, we're going to develop, well, and we say us, we. It's a mission of the CNP and to provide support to research and development in the rest of the country. We expect that Orion and CIRUS, as well as other laboratories, will be open to external users. So we're going to be offering the possibility so that researchers in Brazil and researchers around the world may come and use our facilities for research purposes. The expectation is to develop diagnostic methods, vaccines, discovery of new pharmacological products, and that will be very important for strategic development of strategies, epidemiological strategies. That will be very important for our national public health system, including, and it will boost our sovereignty in terms of new challenges, health challenges like we had one recently. And what is especially unique, what is so unique about Orion that needs to be said? Orion will be the only laboratory at level 4 in the world that will have light lines directly attached or linked to it. What will be these light lines? We'll have 3 light lines for studies of X-ray, MRI, tomography. They'll be called Cibipiruna, hibiscus, and timbó. And with these light lines for X-ray tomographies, we'll be able to study cells up to a level of organelles, and we'll be able to study tissues and organs. And we're talking about X-ray tomographies and 3D studies. And we're going to be able to do studies with living animals. It's a unique capacity. No other laboratory at level 4 in the world has that kind of capacity. It's going to be one more reason for us to be proud of it. Why Orion? CIRUS, I don't know if it's the brightest star in the sky, and Orion is a constellation that is close, and astronomically speaking, is close to CIRUS. And in Orion, we have the 3 Marys, with a beautiful poetic analogy between the correlation between CIRUS and Orion, because we have the 3 lines of light going into Orion and providing us with a beautiful analogy with CIRUS, meeting these 3 Marys, if you will. And finally, talking about the Bioscience National Laboratory, what is our mission? To use integrative biology to understand health and illnesses. And by so doing, we hope to provide support to the development of health solutions for Brazil. We're able to work with living matter from small scale and molecules to organisms. And how can we do that? Because of our expertise in protein biochemicals and biophysics. And that expertise is our core. LMBIO was born as a laboratory providing initial support to our ancient cyclotron source of light. It was a laboratory that provided support to protein crystallography in our ancient source, an old source. That's an activity that we've been undertaking for the longest time. And today we have the ability to work on cells at various levels of complexity, basic cultivations of 2D, 3D, bioimpression, bioprinting, microfilament. We also have a laboratory that works with in vivo models, bioteria, and we have the ability to do genetic additions of these animals. It's an ability that's not very common in Brazil. We have the autonomy to produce these transgenic animals, and of course, computational biology, which today is moving in the direction of using artificial intelligence algorithms to solve problems related to health. We have 5 divisions at LMBIO, 2 divisions that we call Discovery Science. And these divisions of Discovery Science, that's where we do our research. We've been acting, our research groups, a strong focus of our work, research work that we do is to understand molecular mechanisms behind diseases. We're very keen on understanding those disease mechanisms at the cell and molecular level. And they've been working on viruses, bacteria, Chagas disease, and also and there's another division called the Integrative Biology Division, which I lead. We've been doing cancer studies and neurodevelopment studies also, and cardiovascular studies, always using biocomputational research to support this work. We have a division called Advanced Health Technology Clusters, or it's advanced medicine, and in this division we have a pipeline of drug discovery, which uses primarily products from our own biodiversity, natural products for our own biodiversity. We have a park of equipment of mass spectrometry geared to the research. It's very modern and large. We've just installed a new equipment that will be dedicated to single cell research. It will be the first such laboratory able to undertake this kind of experiment. And in this laboratory, proteomics laboratory, the great mission is to deliver and to discover markers and targets for many diseases. And we've begun studies on structural proteomics. We have a tissue engineering laboratory, and it is being moved to another area of tissue engineering. We're going to be able to do 3D cultivation, microfluidity, and we're developing regenerative medicine protocols and genetic research as well. And finally, we're implementing a laboratory of immunobiologicals whose mission is to undertake research and development for immunobiologicals. I'll talk to you now about this laboratory in particular, what we have implemented so far. We are setting it up through the development of a monoclonal antibody. It's a pilot project. We expect that in its full capacity will cater to different projects for immunobiologicals in general terms. Now, excuse me, first of all, I'm going to talk about our 5th division. That's the division of facilities and services, in which we have the coordination of all the laboratories that are specialized at ENG. These laboratories give us the capacity to do expression, purification of proteins, production of biomolecules such as peptides and nucleotides. We have the ability to do the characterization of these protein molecules as well as others through magnetic resonance, MRI, crystallization, spectrometry, and calorimetry. We have the ability to do high content screening, microscopics, through optic microscopy, and in vivo studies, and cellular culture, metabolomic genomics, and our ability for computational biology as well. Now, talking about our pipeline of immunobiologicals, I will emphasize where we understand we can make a big contribution to the research network in Brazil. It is not always that when you have a target to be aimed at in immunobiologicals, you don't always know how that immunobiological needs to be dealt with, and frequently you need to do a design for that particular immunobiological to ensure it will be most effective. Our mission, of course, has been increasingly We have recently received from our general director the information that we need to go into it strongly with artificial intelligence. We've done that already, but that's decidedly will step on the gas pedal because we understand that these algorithms that have been launched are extremely important. We understand that they won't replace totally, but they have the capacity to speed up research in proteins. And of course, when we're talking about biologicals, especially when it comes to proteins, here I just want to bring you information that's food for thought. These data are not ours. They come from David Becker's lab. He is the Pope in the protein design. And this paper came out December last year. And I don't know about you, but I work in structural biology. But now you can design proteins that are not out there in nature around a target. I was impressed by it. This publication came out recently. It's a peptide hormone. They were able to design a ligand, a protein. Again, the protein is not in nature. It was invented by a computer. It has high affinity for that target, with that target, and obviously thinking of the development of immunobiologicals. This peptide can be an antigen, and you can decide you can design better solutions for it. This information that came out yesterday, I had to add it to my presentation. I don't know how familiar you are with these programs, but this AlphaFold came in to revolutionize structural protein molecular biology, and it predicts with high accuracy the spike protein on a coronavirus very accurately. This is a predictive molecule that is very similar to the molecule experimentally seen as the molecule it brings antibodies, and this is the sky is the limit. And what have we done? We have developed protocols to modulate TCR interaction and antigens. It's been done through cooperation to develop vaccines based on T cell, and we have developed projects as well. Given a certain a known structure of antibodies, the computational pipelines will be able to predict mutations that will improve affinity or modify the way to interact the PCR and the antigen and the antibodies, thereby evolving those antibodies and designing new ones. We have just set up our capacity to add capacity once the sequence has been drawn. We hope it's the ideal sequence for the therapeutic target. Of course, we're doing genetic engineering to introduce the sequence inside the cells. You need to select, and now I'm saying something that it's basic for everyone. You have to select the cellular clone with the highest production of this immunobiological. We have just installed equipment to be able to do this the selection of these cells, of these clones that have the maximum production of the immunobiologicals. We're setting up capacity to expand these colonies, and all of these things that's being done in our own new laboratory. It's beginning to operate, and when it is working in full capacity, we expect to provide research cell banks to the community so that they can develop projects together with us, and in the future that will be done in BPL. Well, finally, this is my last slide. This is the area in which we believe we can make our biggest contribution to the immunobiologicals community, which is the characterization of biological complexes or immunobiologicals and the target. And we can help tremendously on the characterization, especially from the point of view of structural biology, because we have our source of synchrotron, and through this synchrotron source, you can characterize this interaction in atomic level using protein crystallography, and you can do characterization from the point of view of solution behavior using small index of spectrum and secondary, tertiary structures, circular prisms. We have in the National Laboratory of Nanotechnology this microscope called Trotoscope. It's a very powerful laboratories. We just installed a laboratory, will be 2 such laboratories in the country, and you can do a technique called electronic cryospectrometry to solve the structure of these complexes in atomic level. And finally, at LMBIO, the park of equipment that we have, we hope to have convinced you that we're able to help in various assays of characterization, functional characterization, and the interaction between biology and target using spectrometry and cellular biology. And from the point of view of biophysical techniques, we have plasmonic resonance, light dissemination, fluorimeter centrifugation, ultra-analytical centrifugation, just to mention a few of them. The capacities are many, and again, I would like to I'd like to draw your attention to our spectrometer center, which is very modern, and we can do the sequencing of protein and most traditional research. That's it, basically. I would like to thank you for the opportunity to talk about CNPEM, to talk about LMBIO. I'm leaving you my email of the director Maria Huda if you're so interested in developing a specific project with LMBIO. Get in touch with her to understand how they can operate. Those who want to use our facilities as external users, put it on Google, CNPN, open facilities, and you're going to get to the page and understand how you can apply it to your projects. Thank you so much.
So thank you very much, Sandra. I would like to pass the floor to Dr. Ricardo de Godoi of Bio-Manguinhos, from Bio-Manguinhos. Ricardo, I'm going to let you know when you went through 12 minutes, okay? Okay, good morning, you all. I would like to thank, first of all, the invitation to be here talking to you, to my colleagues. Sandra, we have a very strong interaction with CNPEM, especially with Bio-Manguinhos. We are assembling a strong collaboration. We had the presence of a good part of our team visiting the facility. So I think you have a great opportunity, and it's becoming clear, as Leda mentioned, and thank you very much for your opening, the growth of the ecosystem, and we have to be increasingly closer to our product, of having our products. So I'm going to go through 4 main topics, and this presentation was prepared by Elaine, who is the chief of department of the pilot project in Bio-Manguinhos, but she was not able to be here, so I will make the presentation too. But I would like to thank Elaine for assembling the presentation. So I'm going to go quickly through a vision of what is Fiocruz to understand the context of Bio-Manguinhos, and also a little bit of the pilot plants and the perspectives of collaboration. So Fiocruz is an institution, a centenary institution. We are present in Brazil and several states. We are present also abroad, and we have an intention of acting intentionally, increasingly intentionally, and this presence also reflects in an actions in several fields. So we act in the production of biologicals, of pharmaceutical products, and also in teaching and several levels with graduation programs, also that copies the program. We also have technical teachings and programs of communication and health. So we permeate several processes in health. So this very singular aspect of Fiocruz brings some opportunities and challenges for the management of this system. And we have a focus on meet the demands of the Brazilian population and national level, the direct relationship with the Ministry of Health, which we are part of, and we also act in several fields of basic research. So this slide, I'm sorry it's in Portuguese, and I know that part of the audience has interpretation, but I was not able to translate it into English. But we have in the sphere, this system, we have Bio-Manguinhos, which is the organizer of this event, and we are more recent, although the activities that we do there are previous to the launching. So we were founded in 1976, and we had our anniversary last Friday, and we have an expertise in technology transfer, research and development, also clinical trials and other fields. So in this context, these are some of our figures that we have for supplies in the last years. And in this context, we have a very broad portfolio from vaccines and diagnostics, biopharmaceuticals, and we're also crossing other frontiers. And our team is here, Hosanna and others that are worked in advanced therapies and cellular therapies. So it's under expansion and a portfolio that brings also challenges. And in this context, we have acted with responses to challenges and public health emergencies. And we today are dealing with some situations regarding the dengue fever and the situation that we are seeing in Rio Grande do Sul state. So Bio-Manguinhos is always responding to these needs, and I think this is part of the momentum that we have, the drive that we have. So we have to work to have less emergency, and this is one of our concerns, and we think that vaccination is fundamental for everything, for these goals. One of the points of the tables is to talk about partnerships, and one of the partners of Bio-Manguinhos that come from its foundation, acting several products. Then we have a very strong background series, and this is of course summed up, but we have important landmarks that show that these partnerships are being carried out in Brazil and also abroad with countries of several continents. And I think that this movement is essential for the development of our activities. So I'm going to talk about the innovation area in Bio-Manguinhos. We have 4 axes of acting. Of course, there are interfaces and other areas that we work with the providing of technological service, but we also have the research and development with our own products, and we also have the process optimization, which is very relevant for those who have a great number of products in its portfolio. So this is an improvement area that is essential in our context, and it demands a great part of our efforts. And also in the sphere of innovation, the technology transfer field, and also the medical area that we called clinical trials, but have many other attributions from the evaluation of new projects. We have to have this standpoint of the relevance of the products and how they're going to be implemented, and this back-to-end vision sometimes lacks from some of our products. Sometimes we have a great degree of effort for breakthroughs, and sometimes we lack of looking at the possibilities of usage. So, and with the growth of the perspective of great breakthroughs, the access to new technologies is growing, and with that, the challenges of we will talk about in the next table, the valley of death, and they're changing the locus and becoming closer to the end user. So this is a topic that is within intense debate, and which is part of another frontier dimension of discussion that we will have to organize with these new infrastructure and with Brazil, and we talk better within these plants and these partners with technological competence so that we are better aligned and the problems that we will tackle. So in the sphere of, we have several platforms and competence of action, and we are able to act in almost every phase of the technological development, and we are advancing to having a better quality in the spheres of our development, and we are advancing to the best practices in the laboratories and the spaces that we act. So we are working in many fronts, and we are seeking to improve all the initiatives, and their new products are being thought of in this umbrella of initiatives, and this is another new frontier that we are overcoming for the new products. So we get to the prototype plant, and this plant, as I have mentioned, it's coordinated by Elaine, and she has a comprehensive experience in this production area and all the challenges that are connected to this. So we have a virtual tour. She doesn't like very much because it's not it's completely updated, but I think it's good for you to visit and for the communication, and so that people get to know it's a clean environment, controlled environment, and has very restrict access. And we took advantage from our communication unit in situ, and so to open this opportunity so that people could visit virtually. It's not as it is shown, it has evolved from this was made, but I think it's an opportunity to make a virtual tour and to know it from the inside perspective. And we also have a degree of capacity of acting in the level of manufacturing best practices and clinical batches production, and we have been working with the production itself. And we have scale challenges when we get to the biological projects. We are used to deal with vaccines that are billion of doses or dozens of billions of doses, and some of the products that we work are of thousands of doses. And this changes very much the way we do things, and it's very helpful to have this facility to work with this finished product. We have three great groups. We work with eukaryotes and prokaryotes and conkaryotes, and a final processing pilot lab where we do the formulation, the fulfilling, formulation, filling, freeze-driving, labeling, packaging, the finishing products. So we work with products that are in the market already, and we work with the new products that we are developing, and we have a possibility of advancing with the new vaccines with mRNA. And the fact that we have this plant, and maybe we will have still 1 year of development and an interaction with a new partner, and we are also able, although it's being developed with the best practices, and we hope it can achieve the best practice in production. So this means new processes and quality assurance. So this experience fitted in this innovation ecosystems is being essential for Bio-Manguinhos, and we hope that with time we are able to adjust and to better use this plant so that we can open it to other partners, and thus be able to expedite not only our own development, but also development of other partners that we have nationally, internationally, and working in this perspective, and we have the installed capacity for the production of new products. And from that was some things that were mentioned today to better talk and discuss with other agents from this ecosystem so that we can accomplish these higher targets and objective to and strengthen the system as a whole. So this was the message that I wanted to pass on to you, and I would like to thank the opportunity to be here. Thank you very much. So thank you very much, Ricardo. We are going to now pass the floor to the presentations.
Ella from Bharat Biotec for the presentation on their structure for vaccine innovation. Thanks, Dr. Ella, for joining us.
Thank you for that introduction. Sorry to switch to English, but I hope you can hear me and see my slides. I'd just like to thank the Fiocruz organization and Bio-Manguinhos for giving me this opportunity to talk to you today about the kind of infrastructure you require for innovating vaccine development. My name is Raches. I serve as the Chief Development Officer here at Bharat Biotech. And before, I thought that I'd frame today's discussion based on a case study. When it comes to innovative vaccines, the most recently manufactured vaccines are the COVID-19 vaccines. And I would like to frame my discussion today in the form of 4 pillars that I think are crucial for innovating vaccines. The first being the vision, what kind of organization you work for. At Bharat Biotech, we are headquartered in India. We are a vaccine manufacturing company established in 1996. We manufacture vaccines and biotherapeutics. And by volumes, we're the 5th largest vaccine manufacturer in the world, where we primarily supply to emerging market areas, that is Asia, Africa, and LATAM. And many of our vaccines are developed in mind with efficacy, safety, equity, and accessibility. Those are paramount important attributes for us towards vaccine development. The second main pillar, nothing can be done in a silo, and collaborations are required. Many of the vaccines that I'll be talking to you today about are innovative vaccines of its own class. And over the last 25 years, since we've been a company, we've established significant relationships with external agencies. These can be academic organizations, government infectious disease research organizations, or also several other companies, as the presentation similar alluded to. When it came to COVID-19, it was a challenging time for everyone, and at that same time, we really didn't know what platform would work, what kind of vaccine would work. Was it mRNA, viral vector, inactivated, or recombinant? At that time, we were not sure. So we started working on 4 different vaccines in various, with collaboration agreements with several organizations highlighted on this slide here. One of the first vaccines we licensed was an inactivated COVID-19 vaccine, which we published significantly in peer-reviewed journals. And we were able to commercialize this vaccine within a year's time, based on the infrastructure we've developed over the years of product development. The third most important pillar is the expertise that the company or the R&D facility needs to have in order to develop such vaccines. The first, under expertise, it's very important to have optionality or the freedom to operate between different platforms. I don't think that one platform fits all kinds of pandemics. I think there is a reasonable case to have multiple attempts at goal. So with that regard, each platform has its own advantages and disadvantages, and it's important for a vaccine manufacturer such as Fiocruz, Butantan, or Bharat to have that availability to pick and choose which platform we would like to use in our vaccine development efforts. With Bharat, we had a significant amount of expertise in viral cell manufacturing. Many of our vaccines are currently licensed with this virocell vaccine for rotavirus, Japanese encephalitis, rabies. We have a phase 3 chikungunya vaccine, a Zika vaccine, which is in phase 2 clinical trials. All of these vaccines highlighted on this slide are inactivated or live attenuated vaccines manufactured with this virocell platform. And it's an extensively studied platform for us. We supplied 300 million doses of vaccines with this platform, and we're really assured of the safety profile of this platform. Another important aspect for a manufacturer or developer to be fully equipped with is its ability to do clinical trials. Usually you need to do clinical trials where disease is actually prevalent. Some of the diseases that we work towards against are not necessarily in India, they're mostly in Africa. Hence, we have to do phase 1, 2, and perhaps phase 3 efficacy trials in the different parts of the world. So it's very important to have that translational ability, not only in your home country, but also outside of your jurisdiction. And since if you're making an innovator vaccine, these vaccines are primarily going to be licensed on efficacy or effectiveness readouts. It's not going to be an immunogenicity or an immune response comparability study. Hence, it's very important for a developer or a manufacturer to have an ability to generate efficacy or effectiveness data wherever that disease is prevalent. At Bharat, we've embarked upon several phase 3 and phase 4 studies. These are largely powered sample size clinical trials in close to 10,000 to 30,000 subjects. And these are all looking at efficacy and effectiveness readouts, be it for chikungunya, cholera, rotavirus, typhoid. We've done a fair share of these kind of clinical trials all across the world. The last main important pillar I would like to talk to you about is the skilled manpower. It's very evident that there are very few vaccine manufacturers in the world, a few noteworthy in Brazil, a few noteworthy in India, and the rest of the world as well. It's primarily because it's a very difficult product to make. If it was that easy, you'll probably have a lot of manufacturers. One of the reasons why we were able to work on an inactivated COVID-19 vaccine is our ability to have a BSL-3 production facility. BSL stands for Biosafety Level 3 production facility, where essentially highly contained viruses can be propagated and grown in these state-of-the-art facilities. As you can see here by pictures of my colleagues during the pandemic, were handling live virus in these labs. And essentially this vaccine that we developed involved growing the virus at large scale, about 10,000 liter bioreactors. That's the scale at which we were growing these viruses in viral cells, and then inactivating them with a chemical agent. Another novel part of vaccine development is the adjuvant development. As you know, adjuvant is a molecule used in a vaccine to trigger an immune response or boost the immune response. And traditionally, there have been only a select few adjuvants that have been licensed commercially. Algel is also known as aluminum hydroxide. It's a very commonly used adjuvant, but it does have a few weaknesses. The first weakness is its inability to generate cell-mediated immune responses. So you really don't have too much of T-cell activity if you use alum. But what we did at Bharat is we tweaked our alum formulation to include in TLR7, TLR8 agonists that led to a balanced immune response. So not only we were able to see neutralizing antibody responses, but we were also able to detect cell-mediated immune responses, which is very important for vaccines to have, to provide short-term and long-term protection. Another important pillar that we had during this process during the pandemic was our ability to collaborate with labs, external labs. These are government labs that could do live viral challenge studies for us. For the inactivated vaccine, Covaxin, I mentioned that we did 2 live viral challenge studies, one in hamsters and the other in non-human primates. Both of these studies gave us the confidence that this vaccine was protecting in animals and could show a beneficial protection in humans as well. So it's a difficult study to do because you have to vaccinate animals to placebo or vaccine, and then you need to challenge them intranasally with live SARS-CoV-2 virus. So select few labs can only carry out these studies. And during the pandemic, it was a very difficult time to actually get these labs available for your vaccine clinical development efforts. And then with this vaccine, we were able to, after the 2 live viral challenge studies, we were able to quickly progress to a phase 1 and a phase 2 clinical trial. Phase 1 and phase 2, as you probably are well aware, you're primarily looking at safety and the immune response in small sample sizes. Where we had to go to larger sample sizes in a phase 3 was to evaluate efficacy of this vaccine, which was reported in a 25,800 sample size clinical trial. We reported a vaccine efficacy of 78% against moderate disease and 93% against severe disease. And this was all done during the Delta variant that affected India terribly during 2020, late 2021. Part of that infrastructure that is so crucial is your clinical trial infrastructure. And the timeliness of doing it, especially in the pandemic, timelines were crunched, not only in product development, but timelines were also crunched in clinical development. And as you can see here, these are some facts from the phase 3 clinical trial that we conducted, where many aspects of a clinical trial, planning of a clinical trial and conducting, which required anywhere between 1 week to 20 weeks, were all crunched down to 1 week to 2 weeks. For example, we conducted a site initiation visit in one site on Monday, and on Tuesday we started enrolling and dosing them. So that was the speed at which we were operating with. And of course, this can't be done only with one vaccine manufacturer. You need to have capable CROs. You need to have capable institutional review boards or ethics committees to ensure that these clinical trials are done at a rapid rate. I'll just like to end by talking about another interesting vaccine that we've recently licensed. It's an intranasal COVID-19 vaccine. And this stems from our interest that the parenteral vaccines, or the vaccines that you inject, fail to prevent infection and transmission. And that was one of the points that stuck with us during the pandemic. All the vaccines that were licensed in the first wave of COVID-19 were all injections. And as you know, they probably failed to prevent against infection and transmission. So we started collaborating with this university at Washington University in St. Louis to develop an intranasal COVID-19 vaccine in the hopes of it being beneficial against infection and transmission. One of the prime reasons why we selected to work on this vaccine is our collaborators working that validated this vaccine in 3 different animal challenge studies. So it's the same live viral challenge study that was done here in mice, hamsters, and macaques. That gave us a reasonable amount of evidence that this vaccine was able to not only protect in the lower respiratory tract, but also protect against infection in the upper respiratory tract. Once we technology transferred this vaccine into Bharat, we were able to scale up the vaccine, and we were able to conduct several phase 1, phase 2, and phase 3 clinical trials, ultimately leading to licensure based on comparable immunogenicity readouts between an injectable vaccine and the intranasal vaccine. We are still trying to evaluate this vaccine, how well this vaccine works against infection and transmission, and we're exploring different kinds of clinical studies. One can also think about human infection models for SARS-CoV-2, which is being evaluated at the moment. To summarize, I think it's important to have, based on the 4 pillars I mentioned on the vision, the expertise, and the collaboration, and outside of product development and vaccine development, where you need to have the resources, infrastructure, and facilities, to manufacture a vaccine candidate, what's gaining more and more importance as we go along this pathway of vaccine development is your ability to do robust preclinical studies, not only preclinical toxicology and potency studies, but also potentially efficacy studies in animals, in preclinical. So these are primate studies, it could be hamster or mice efficacy studies are gaining more and more importance. Clinical trials have become more adaptive and operational seamlessly conducted. One can also start looking at how human infection models, or CHIM models is what we call them. One can also look at how well these studies can be integrated into your clinical development plan. And once the vaccine is licensed, I think that, I mean, there's still work to be done. You still need to be looking at post-marketing safety surveillance studies on these vaccines. And you need to also look at how well these vaccines, at least with COVID, how well these vaccines are working against variants of concern or variants of interest. So it's a broad scope, and I don't think it's only R&D centric. There is a translational side to it that is gaining importance, and there's also a post-commercialization phase that is gaining importance to validate the safety and the efficacy of your products. Thank you. I'll be happy to answer any questions provided this time.
Thanks, Dr. Ela, for the excellent presentation. I'll switch back to Portuguese. Então, nós temos pouquíssimo tempo para perguntas, mas vamos abrir para cerca de 2 a 3 perguntas, idealmente uma para cada palestrante.
Each lecture, ideally. I can see here that online, I still haven't received any questions. Please, go ahead. Good morning, everyone. Leda, I'd like to congratulate you on the excellent work during the pandemic with the development of potential Brazilian vaccines against COVID. Ricardo, Sandra, our colleague from India also. I'd like to congratulate all 3 of you for your brilliant presentations. Sandra, as a researcher, I see a dream coming true at last. And as a citizen, we have to be proud of everything you've done. And now thinking of sandwich scholarships in terms of research at copies, I'd like to know 2 things. The managing board, how does it work to prioritize the projects that get to you? How do you go about selecting projects? And in addition, how can we get our students, for example, thinking initially of CAPS courses, for example, instead of going abroad, it's also important, but they should be developing part of these projects here in Brazil. And I'd like you to talk about those 2 points, if you will, please. Thank you. Sorry, I forgot your name. Joseph. All right, Joseph Paul from ISC and CNPQ. Thank you, Joe. Thank you for the great question. Well, we, as I said before, initially, we have this large mission of offering several laboratories that we have, the facilities to the community of users in Brazil, academic users and corporate users as well. And the way these projects are selected ends up being a bit within the format of each laboratory. Each laboratory operates in a certain way, and it's a distinct manner. And for some facilities, basically, you apply for a project, and you have to do a selection, and that has to do with the complexity of the facility, the number of proposals that are sent. So for the synchrotron laboratory, there are committees geared to assessment made up of internal researchers as well as external users, and these committees are specific to each line of research. Projects are sent, they're assessed, and as a whole, and it's a comparative analysis are selected, and we do the publicizing of these projects. At NBio, we have laboratories that are open to users. We want to expand them briefly. We will have another 4 laboratories or 6 in a total of 10 laboratories. And again, each laboratory will have its own dynamic depending on the flow of projects that will be sent to them. How then do students in Brazil and the universities in Brazil, how can they access our capacities and facilities and develop their projects? One of them is to apply for projects, and then this model of student that happens. It's been happening for several years. CNPEM operates for over 20 years, at least 25 years, I would say, and since our first look from source of light, we've been catering to students from many institutions in Brazil and abroad throughout these years. And to operate projects in a more complete way, we also have models where, through partnerships and cooperations, students come to our institution and they can spend months undertaking their research projects, and that ends up being at the discretion of different research groups which work together. It depends on the context that are established. In LMBIO, under the new leadership, the expectation is to issue calls for projects in the sense that these projects will be carried out almost entirely at the laboratory. That's what we hope to have in the future. Thank you, Sandra. We have the time for one more question. Congratulations for the great panel discussion. My name is Renato Estrella. I'm from the Butantan Institute, researcher. Ricardo, I'd like to know, in your pilot plant, the pilot plant that you have, we're thinking of a business model now. I think that you're planning to have, do you plan to have partnerships or services? And how can you compete with these services, external services hired abroad, vis-a-vis costs and speed? Because we know that the speed here for us to acquire inputs for new projects, that could be a hindrance to us. It could be an obstacle. How do you overcome that obstacle? Have you had it mapped out? I'd like to draw on this opportunity to say that Paulo Lebo's right there at this point, at this moment. That's why he couldn't make it today. Deal with a partnership that we're trying to establish and exchange information on the production of RNA. We're developing this model literally now with you, and what we first of all we're trying to stabilize the internal use. We are a relatively new facility, a young facility, and starting through a strategic interaction to receive our science and technology projects. We're launching this model with Butantan literally today. Paul is there. He has Eleni Patricia with him, the coordinator of the project, and we will be seeking out a qualitative differential. We've done improved process improvements and the development and the manufacturing of our prototypes of mRNA, and that's how we're going to be trying to gain ascendancy and building a technological differential that's innovative in the production process. We have the first facility, GNP, it's in its final phase of engineering implementation, a new platform that will do the automated production, and that's how we're going to seek out that new route. We're not trying to, first of all, we're not seeking competition in terms of volume or financial sums. That's how we're going to develop the relationship with Butantan. As of today, we're going to sort out the main issues, but the initial focus will be on strategic projects for the country and the organization as a whole. I think we're materializing this project today. Hopefully next year, we'll be able to tell you about this whole story in more details. Thank you for the invitation. Renato, perhaps I could contribute to what you just said. During the pandemic, we looked for budgets from many CROs abroad for different parts of the development of vaccines. And we know that the costs are staggering, millions of dollars, and that's and stuff that we ended up doing in Brazil for a small fraction of those costs. So that there's huge potential. And also we have shared with Elena Caridi and other researchers, and we're able to find the certification of cellular banks, and we obtained bio-reliance certificates, and we found a network in Brazil with several laboratories that could do, at least in a phase 1 of clinical studies, at very low costs, and there was a line abroad. So it's a matter of time. We were able to do it at much lower costs and reduced time frame. We were able to do things that are acceptable from a point of view of the meetings we had with Anvisa and so on. And I think that that's the question of following what Ricardo said, trying to come up with a network among these institutions, bringing them all together. Our pharmaceutical industry does a great deal of outsourcing. Even large biotechs in the world, large pharmaceuticals in the world, they do outsourcing of various R&D stages and development and research. LMBIO presented what we can do for Bio-Manguinhos as well. I talked about the many institutions that are being commented upon.
Biotec, os NB3 para fazer estudos de desafio, para ver eficácia de vacinas. A UFRJ tem um NB3 multiusuário. O segundo criomicroscópio que tem no Brasil, que você falou também, é multiusuário na UFRJ. Então, para além do nosso laboratório, da nossa planta piloto, a parte de caracterização, de ensaios de desafio também, a UFRJ aí está disponível. E eu acho que a gente tem que, então, seguir. Vou finalizar com essa mensagem que, na verdade, veio de um cochicho ali do Ricardo, né, que a gente tem que fazer uma rede dessas instituições que podem, a nível de Brasil, trocar experiências e encontrar sinergias para que a gente consiga desenvolver cada vez mais a área de biológicos no Brasil, porque desenvolver a área de biológicos no Brasil significa reduzir custos para o SUS e aumentar o acesso da população a esses modernos medicamentos e vacinas da biotecnologia para a saúde humana. E acesso da população à saúde, né, é o que tem que ser o que nos move, né? Então, queria agradecer muito a participação de todos, né, e finalizamos assim nossa mesa. Obrigada aos palestrantes.
Obrigada, Leda, pela sua moderação. Obrigada também aos palestrantes. Pausa para o coffee break, mas não deixem também de assistir aos posters e visitar os estandes. Tá bom? Voltamos em 30 minutos. Até já.